All right, so in the previous video, we talked about the structure of DNA and how at its core, it's made out of nucleotides. There are 4 different versions of those nucleotides based off of the nitrogenous bases, so A, T, C, and G. We talked about how A and T will always pair together and C and G will always pair together. And when these nucleotides come together, they create this double helix shape of DNA. So in this video, like the slide says right here, we're going to be talking about DNA replication. So when something replicates, it makes another one of itself. So again, if we wanted to like replicate like a piece of paper or something, we would just copy that piece of paper to make it look identical to the original copy. So in this case, we are going to be copying DNA. Like I've said in the previous video, you need a complete copy of DNA in every single one of your cells in your body. So when you make new cells, which we'll get into in the next video, but when you make new cells, your new cell needs a complete copy of DNA, which means we need to first make a copy of that DNA. We need to replicate that DNA so that we have a copy to stick into that new cell. So we're going to be looking at the process of how DNA actually replicates itself so that we do have that new copy to stick into our new cell. So we say that DNA will replicate itself based off of the semi-conservative model of DNA replication. So let's break down this word semi-conservative. Conservative means that you try to keep things the same. So maybe if I wanted to, we can talk about like conserving energy or something of that nature. You're trying to like limit or keep the same how much you're using. Semi means it's kind of conservative. It's kind of the same. So let's look at what this actually looks like with DNA. So what you're seeing here in this diagram is our template DNA, our original copy of DNA. Now, like I said, we need to replicate this DNA because we need to make more cells. The new cells we're making need a complete copy of DNA. So what we're going to do is take our DNA right here and take our 2 strands and pull them apart from each other. We basically unzip the DNA so that we take our double helix or our double-stranded DNA. We separate those 2 strands so we have 1 strand of DNA and a second single strand of DNA. Now, because we can use those 2 separate strands as templates, because it has the A's, T's, C's, and G's, we're able to create complementary strands based off of the base pairing rule. So what you're seeing here is that we've taken our double-stranded DNA, we've pulled it apart. So that's what this blue is right here in the double helix, and here's the blue over here in the double helix. And we've used both blue strands as a template strand, and then we were able to take new nucleotides and create a complementary strand based off of the base pairing rule. We are going to see some videos on this if this is a little hard to visualize in your head, but basically on your DNA strand right here, if it goes A, T, C, you know that the complementary strand over here, if it goes A, it's going to pair with T on the complementary strand, T is going to pair with A on the complementary strand, C is going to pair with G on the complementary strand. And that's how we're able to create that second strand to create 2 double-stranded DNAs. That's what you're seeing right here. Now, DNA can go on and replicate itself again if we need to make more cells. So again, you're going to see that these 2 strands are pulled apart from each other. Sadly, this isn't color-coded great for this second generation of DNA down here. But again, we are conserving our single strand of DNA with the blue strand. We're creating a new strand of DNA, a complementary strand of the pink strand. And you will see the same thing in the middle here. It's just they're using pink for both of them. But one strand is conserved by the template strand that you're getting up here. And the other pink strand is going to be your complementary strand. So again, same thing here. We take our blue strand, we create our complementary strand in pink. We take our pink strand up here, bring it down here, create a second complementary strand still in pink. Now, that's sort of the basic gist of how DNA replicates itself. We are going to have to go into more detail of how this process actually happens. So that's what this diagram is showing us right here. And I do have a video to show what this looks like afterwards as well. So what is going on is this yellow part right here, this is our original DNA. This is our template DNA. And what we've done is we've unwound the DNA and we've pulled those two strands apart from each other. Now, over here, we use an enzyme called DNA helicase to unwind and unzip the DNA. You don't have to remember that word, I'm just pointing it out. DNA helicase is going to continue to move in this direction to continue pulling apart our two strands of DNA. Now, keep in mind, both of these yellow strands are considered a template strand. Both of these green strands are considered complementary strands. However, the way that DNA is replicated, it creates two different ways of replication. So right here where the DNA helicase is, this is your replication fork. Because we get a fork in the road with the DNA. And then we have two different styles of replication. So we have a leading strand style and a lagging strand style. Now for the leading strand, and this has to do with the structure of DNA, when we're creating our complementary strand, DNA polymerase, our enzyme, will just add in your nucleotides that complement your template strand, and it moves towards the replication fork. So it's just gonna like zip on through, just like adding in its nucleotides until it reaches the end of the DNA. The lagging strand is a little bit more complicated, and again, this has to do with the structure. Nucleotides can only be added in one direction. We call it a 3' and a 5' end of the DNA. Don't worry too much about this, but we can only add nucleotides moving in the 5' direction, so that's why it can only be added in one direction. So in this case, we have to add a complementary strand in segments. These segments, so here's one segment right here, here's your second segment right here. These segments are called Okazaki fragments. There's a Japanese guy, he discovered the lagging strand way of DNA replication, so he named it after himself. But what you're seeing here is that when the replication fork was over here, we came in and we started adding nucleotides going away from the replication fork. But as the replication fork continued to move along the DNA strand, it opened up a new segment right here. So we added more nucleotides, again, moving away from the replication fork, and they'll be added until they reach the first segment over here. So like I said, I'm gonna put on a video because that will make more sense for you to see it than just listening to me explain it.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So hopefully that video gave you a really good visual representation of what this DNA replication actually looks like on that microscopic level. Because remember, this is happening inside the nucleus of every single one of your cells whenever we need to make a new copy of DNA. So what we're going to do right now is practice replicating some DNA or finding the complementary base pairs to the DNA sequence. All right, so here we go. Here is our template strand up here. I have this purple thing going. We have TAC, AGG, all this stuff. These are our nitrogenous bases for each nucleotide. So the T represents one nucleotide, the A represents another nucleotide, but we're representing them through their nitrogenous base. Now we need to create the complementary strand. So if we're replicating the DNA, we have our template strand, we have our complementary strand. We need to match each nitrogenous base or each nucleic acid with its complementary pair. So I have a little reminder down here. I know it's blocked by some of the stuff popping up, but we know T is always going to pair with A is always going to pair with T, and C is always going to pair with G. So, they're not matching, but whatever. So that's what we're getting here. So T pairs with A pairs with T, C pairs with G. So on the complementary strand, we know that if we see TAC, the complementary strand has to read ATG. And we can do that the same for every single one of these nitrogenous bases or nucleotides. So AGG is going to pair with TCC, ATC is going to pair with TAG, so A pairs with T, T pairs with A, C pairs with G. TTA becomes AAT, CGT becomes GCA, AAG becomes TTC, TTC becomes AGG, GTA becomes CAT, and ACT becomes TGA. So this is a little, I guess, example of how we can base pair these nucleotides together. Now, we're going to do the same thing for number 2 down here. We have our template strand. Of course, it's going to look very similar, a string of letters made out of A's, T's, C's, and G's. We have our complementary strand if we need to replicate the DNA. What I'm going to ask you to do right now is actually pause the video and just try out creating the complementary strand on your own to make sure you understand how to do this. And then afterwards, you can press play again and just check your answers. So again, please pause the video, try to create the complementary strand based off of the template strand right here. So again, T pairs with A pairs with T, C pairs with G. So there's your first one right there. But continue this along, see if you can create that complementary strand, make sure you understand how to do it, and then come back and check your answers after. No, really, pause the video. All right, so welcome back. Hopefully you paused the video and tried this out yourself. Let's check your answers right here. So what we see here, TAC becomes ATG, CCC becomes GGG, GTG becomes CAC, ATG becomes TAC, CCA pairs with GGT, TGC becomes ACG, AAG pairs with TTC, TAG pairs with ATC, and ATC pairs with TGA. So at this point, we are again going to stop the video here. Please make sure you are checking in with your vocab notebook and you are filling that out as we go. If you did have trouble creating your complementary strand based off of your template strand, this is your reminder to please email me or ask during class. But this is a skill you need to understand how to do. So if you are confused about this, please make sure you are finding the help so that you do understand how to do this. Other than that, like I said, check in with your vocab notebook, fill out the slides that you can, and we will be coming back to talk about cell division.